Hello, everyone, and welcome to today's live broadcast on The Road Ahead, What's Next for Host Cell Protein Analytics. I'm Shannon Stoltz for BioPharm International, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by BioPharm International and sponsored by Cygnus. Cygnus Technologies, part of Maravai Life Sciences, is the biopharmaceutical industry's partner in analytics for host cell proteins, other process-related impurities, and viral clearance. Cygnus helps companies developing therapeutic proteins, vaccines, antibodies, plasma derivatives, and gene therapies to ensure the safety of biotherapeutics prior to human trials, regulatory approval, and commercial release. We do have a few quick announcements before we get started on today's webinar. So first, it's going to be designed to be interactive, and we encourage you to ask questions throughout the event. You can submit your questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can also enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. And keep in mind, please, that these slides will be advancing automatically throughout today's event. Otherwise, if you have any technical problems viewing or hearing the presentation, please click on the question mark help widget in the top right of your presentation window. I'd now like to introduce our speaker for today. We're very pleased to be joined by Eric Bishop. Eric is the Vice President for Research and Development at Cygnus Technologies, and he leads the research and development team and is responsible for new product development and custom development services. Eric has over 25 years of experience in host cell protein analytics. Prior to joining Cygnus, he worked for AstraZeneca, formerly MedImmune, from 2002 to 2010. And during his time there, he had multiple roles, from HCP assay development and tech transfer to being the analytical representative on CMC teams, guiding projects from IND to BLA submissions. Thank you so much for joining us today, Eric, and please get us started.
Great. Well, thank you for the wonderful introduction, and thank everybody for taking time today to come and spend time with us to talk about host cell proteins. As she said, today we're going to talk about the road ahead and what's next for host cell protein analytics. So most companies are familiar with Cygnus and the fact that we sell generic host cell protein kits. But I do want to spend a few minutes just kind of talking about Cygnus because we do a lot more than sell generic host cell protein ELISAs. So you can see we do have those. However, we also sell and develop custom HCP ELISA kits for stage 3 to marketed products. We also consider ourselves the one-stop shop for all process-related impurities. So we do carry ELISA kits for all purification leachates, such as protein A and other AAV affinity ligands, as well as some BSA, insulin transferrin, and anything that could be considered process-related impurity. Finally, our custom orthogonal services. So we do offer a lot of coverage analysis, as well as in-depth mass spectrometry as far as understanding and controlling HCP, which will be a lot of the focus of today's talk. Cygnus has been in this field for over 25 years. I personally have been doing this for over 28, and I've been a member of the Cygnus team for over 16. During that time, we really have developed a very deep understanding of all process-related impurity testing, but specifically host cell protein. And there's not a lot of challenges or anything that you could experience in your organization that we haven't experienced before. So we're really trying to be a wealth of knowledge to help you ease your way through your process development, as well as any kind of regulatory submissions or regulatory questions. We have developed a lot of credibility through these years as well. When Cygnus first started, we were a very small company, working typically with smaller host cell or biopharmaceutical companies. But over time, we have demonstrated our ability to endure, but also that we are the experts in this field, and we have become known as the gold standard globally. And during that time, we've also spent a lot of time, not just with industry leaders, but also with regulatory agencies. We have good relationships with most regulatory agencies globally, and that we try to meet with them as often as we can. And companies know that when you partner with Cygnus, you should have a much easier time getting through regulatory approval because, again, we are the trusted brand and the global gold standard. And just a few examples of our experience and trust that we've developed is we have done over 600 AAE experiments with 2D page or mass spec for coverage analysis. Again, this is a proprietary method developed by Cygnus and has been really well adopted and accepted globally. We also support over 200 process-specific host cell protein ELISAs. And again, that's just demonstration that companies and regulatory agencies trust us enough to let us support their phase 3 and post-marketed products. And then finally, Cygnus kits are it's actually 29 of 29 approved CAR T and cell and gene therapy products that have been approved in the United States and Europe. Okay, so the talk today is about where we go from here. But I think anytime you have these kinds of conversations, it's really important to understand where we started. So this slide is a trip down memory lane. Again, I feel I'm pretty uniquely qualified to have this conversation. And the fact that I developed my first host cell protein ELISA in 1998. And back then, we weren't too concerned about host cell proteins. The industry really saw them as more of a process monitoring tool, so that anytime you ran your purification process, you could see relatively the same level of clearance at each purification step. And if something changed, that was a way to flag your process that you could potentially have some type of failure. So because of that, it was almost a wild west as far as the methods that were being used. You can see a list here of some of the pretty basic methods. A lot of companies were using gel-based methods. Some were using the ELISA. But again, there really wasn't any kind of standardization. In 1999, we had Q6B. That was the first attempt at some guidance as far as product as well as process-related impurities. And there's a few things that are important about that document. One, just to kind of lay the ground of where we were at the time, it essentially says that you should control process-related impurities to the best of your ability. But if you do have really good control over them, it may not even be necessary to have an acceptance criteria. But more importantly, throughout that document, it really reiterated the fact that, it was 1999, and it was understood that our industry was in its infancy. So it says, use the best analytical methods that you have available, but as better methods become available, we encourage you to adopt them as they are available. So it really just kind of laid out that, yes, we know what methods we're using now. We're using them because it's the best that we have, but we know that the industry will evolve, and as those methods evolve, you should do so as well. By 2005, we kind of knew a little better. Process-related impurities, specifically host cell proteins, started showing up, causing problems with immunogenicity in patients. So in 2005, there was a white paper called Defining Your Product Profile and Maintaining Control Over It, Part 2. So this was a paper that was derived from a CMC strategy forum of a lot of thought leaders in the field. And they came up and kind of just compared notes and kind of said, what each of the companies are doing and kind of put their heads together. And there's a few interesting things that came from this paper. First was, that it's probably okay to use a commercial or generic host cell protein ELISA as long as it was well developed. It also removed the possibility for using purification process for derived assays. But the two big points that came from here, one is the mysterious 100 ppm that we kind of hold ourselves to as an industry for host cell protein. Again, it wasn't from a regulatory agency. It was a group of industry professionals who said, we generally see things less than 100 ppm for host cell protein, and that kind of stuck. But more importantly was the expectation from regulatory agencies to do a better job with control of host cell protein. And at that point, you were required to do a host cell protein ELISA, but also required to demonstrate coverage and demonstrate that those antibodies are broadly reactive to the host cell proteins that are in your process. Okay, so a lot has changed even since then. So let's look at some of the current regulatory guidance or guidelines that pertain to host cell protein. I mentioned Q6B already. That was our first attempt back in 1999. There's a few along the way here, like ICH Q2R2. That's just validation of analytical procedures that kind of helps you guide you toward validation of host cell protein ELISAs. I think the ones that really stick out here that are really important for host cell protein are Chapter 1132 from USP, which is a general guidance document for everything host cell protein. So what are they, how to make good antibodies, how to make an ELISA, how to do qualification validation. European Pharmacopoeia 2.6.34 is a very similar document. That is just the European perspective on the same topic. And then recently, the USP updated to Chapter 1132-1, which specifically addresses host cell proteins and mass spectrometry. And again, a good bit of the conversation today will be around that data that derives from that chapter. Okay, so let's talk about host cell protein immunoassays and host cell protein analysis by LCMS. So it's important to understand the positives and the limitations of the host cell protein ELISA to really appreciate why we need to do all of this other work. So ELISA is the gold standard for process monitoring and product lot release. It has been, and it probably will be for a very long time. The primary reason is it's the only tool we have that can detect extremely low levels of host cell protein in the presence of very high levels of product protein. If you have a very well-developed host cell protein ELISA, again, developed by experts, it typically doesn't need to be run by experts, unlike a mass spectrometer. So it does require a very high level of expertise to develop. It's a very complicated, complex assay, but if done well, it doesn't require high level expertise. These methods are very easy to transfer, very reliable and robust. So that's great, but the ELISA isn't without its limitations, and this is kind of really the point of why we need to have all this additional information. So the first one is when we do an ELISA and we read the results at the end of the assay, the plate reader or the software is reporting those results in nanograms per mil, which is a mass measurement, but we're not actually measuring mass with the host cell protein ELISA. We at Cygnus think of it more of host cell protein immunological equivalence or equivalence. And I say that because we're have an assay where you're measuring literally thousands of individual host cell proteins. So in our standards, at each dose, we know approximately what the total host cell protein is there, and it gives some level of color. We can use those to generate a standard curve and report the signal from the unknowns based on the color development. Unfortunately, the host cell protein ELISA doesn't tell you anything at all about what host cell proteins are there, which ones the assay is reacting to. So again, it's not an actual mass measurement. It's just an immunological equivalence relative to the total host cell protein standards. It doesn't tell you exactly what host cell proteins are there, as I just said, and then no information regarding which HCPs are detected. So it's really important to have additional information to be able to supplement the ELISA to make sure that it is measuring at least most of the host cell proteins that are present in your samples. So let's talk about mass spectrometry and some of the opportunities and maybe some of the limitations around the methods. So the LCMS method is broadly applicable in the fact that you can apply it to all the expression platforms. It's not dependent on the animal immune response, so you don't have to worry about antibodies. So again, it makes it completely orthogonal to the host cell protein ELISA. You can identify and potentially quantify individual host cell proteins. And also, it's been something that we wanted to do for a really long time. And I'll kind of show some of the examples of why we've had challenges in the past. But another part that's really becoming very exciting is just the rapidly evolving technology. So sample preparation has become a lot more simple. The instruments, the sensitivity, the speed is becoming very user-friendly. And the software improvements facilitated by AI makes all the data analysis on the back end much more manageable. However, just like the ELISA, it is not without some limitations or challenges. A big one being the variable elements affecting LCMS, which is digestion, ionization, intensity, fragmentation. What that really means is that all these proteins act differently, whether through digestion, ionization, how they fly, which can make quantification based on a general standard very, very challenging. The units of measurement are typically in moles. So again, it's kind of hard to put that into terms that we're all used to dealing with. And then historically, there has been a limited dynamic range of the instrument, and the sensitivity and the accuracy has definitely been a problem that we've been working very hard to overcome. So let's start talking about some of the fun things that we can do with host cell protein coverage, really with AAE and mass spec. So as I mentioned earlier, we decided in 2005 that you had to do coverage analysis to justify the use of your ELISA. And at the time, again, the only method available was a 2D Western blot. So we started using that as well. And again, with the emphasis on it was the only tool that was available, not that it was a good tool. We started doing it at Cygnus, and we found that due to the pretty severe limitations of the method, it really wasn't truly predictive of how the antibodies performed in the ELISA. So in 2013, we developed our antibody affinity extraction, and we'll spend the next few slides going over how we're doing that using that for coverage analysis. And we'll also talk about how we kind of extended that out and some of the other interesting things that we can do with AAE. So first, just a general schematic of how this works. First, we make a column using the antibodies that we're going to use in our host cell protein ELISA. And then for coverage analysis, we'll start with either a null or mock transfected harvest sample. We'll take a section or an aliquot of that sample, and we'll set it aside for later. Then the remainder of the sample will pass over the column, the AAE column. We'll wash it till the signal returns to baseline, and then perform an elution. Generally, in order to get enough host cell protein for our downstream analytics, we typically do 4 cycles of this column. At the end of the 4th cycle, we take the elution fractions, we pool them together, we concentrate and buffer exchange. So at the end of this experiment, we end up with 2 samples. The first is the pre-AAE. This is all the host cell proteins that are in your harvest. The post-AAE are all the host cell proteins that the antibodies have specifically pulled from that sample. In the case of coverage analysis, so we didn't have 2 options. The first, and what we did originally in 2013, was detection by large format 2D silver stain. So again, the immunological reaction has occurred on the column, so we just need to visualize with the 2D gels. And then more recently, and most of the time now, detection with LCMS. And I'll kind of go through some of the example data and show you why we have moved almost everything to our LCMS platform. So here you can see examples side by side of panel A, which is 2D silver stain, and panel B is AAE mass spec. So you can see with even in panel A, there are a large number of spots and the coverage is very high, but it really doesn't give you any real information other than the actual coverage. You can see in panel B, this is our mass spectrometry data. So in mass spectrometry, we not only identify each individual host cell protein, but we can determine its theoretical molecular weight and PI and use that to generate these virtual gels. And these gels give you so much more information than you can get just from a normal 2D SDS page. So in this situation, all of the green spots are proteins that were identified in the pre and the post. So those are matched. The red they're only in the pre, not in the post, suggesting that these are the host cell proteins that we could have missed. But it's also important that you can look at these and see that they're fairly evenly distributed throughout the total protein. This helps you feel comfortable that your antibody generation was not specifically biased against a particular type of host cell protein. So you can see this even distribution suggests it's just random missing. And then finally, the black dots are post. So these were identified only in the post, but not the pre. These host cell proteins were obviously in the pre, but the enrichment of the AAE has identified proteins that we would have had no insight to without the enrichment by AAE. So here's the real power and the real advantage of using antibody affinity extraction followed by mass spectrometry. At Cygnus, we often say that all host cell protein are not created equal. There's thousands of individual host cell proteins. Many of them are going to be pretty conserved. It could be low molecular weight, low abundance, and really won't cause too much problem. But there are some host cell proteins that we know to be extremely problematic. And here on the right-hand side of the slide, we have a list of 41 of these individual problematic host cell proteins. And you can see in the left column there under pre are those host cell proteins present in this particular harvest. And the next two are where we're doing bridging of our 550 and 550-1 to see if those host cell proteins, if those antibodies detected those problematic host cell proteins. So when you generate data like this, it can give you, as well as the regulatory agencies, a much level or higher level of confidence that the assay that you're going to use is appropriate for your process. So again, it's lovely to report high coverage numbers, but if you can supplement that and say, I have a high coverage, but I also know of all the problematic host cell proteins, my antibody will detect all of them. So they don't have to feel uncomfortable that you might be missing very important host cell proteins. So again, just a way to really improve your comfort as well as the agencies that you have selected the appropriate host cell protein ELISA. Okay, so let's talk about some of these problematic host cell proteins and how we can identify them in drug substance samples. So I will say that this is something that is relatively new to many companies. Because we talk to them about, it's like, okay, well, coverage is a requirement. It's really good to understand your coverage. But what's really important to understand are the host cell proteins that make it through your purification process and are found in your drug substance. And often customers will come to us and say, hey, that's not a regulatory requirement, and I don't know why we should do that. And I really point out here that regulatory requirements or expectations should be viewed as a bare minimum. This is the minimum work that you need to do for us to feel comfortable that you're using the right method. However, it's in the company's best interest to fully understand and control HCP to the best of their ability. So here's a list of some of the problematic proteins that we've come across that can really lead to a lot of headaches down the road. For instance, we probably have a call once a week from a customer who is 3 to 6 months into a stability study and find that they have complete degradation of polysorbate. And typically when we look into it, it's caused by a lipase in the drug substance that they were not aware of. So again, if you can identify early the problematic host cell proteins that are found in your drug substance, you can mitigate that risk before you get 3 to 6 months into a stability study or before you get into the clinic and have an adverse event. So again, using these types of methods can be extremely beneficial to your long-term success, regardless of what the regulatory expectations are. So here's the schematic of AAE for drug substance. It's exactly the same as far as how it works, but you will notice that the only option for the analysis is by LCMS. You really can't use any gel-based methods anytime there's product present for a lot of different reasons. And as I said before, we've been wanting to do mass spec analysis for product containing samples for many years. However, the image on the left is kind of a good illustration of why that's been a problem. If you just inject drug substance onto a mass spectrometer, you will see a tremendous amount of product-related peptides from the heavy and light chain. But again, due to the abundance of the product relative to host cell protein, you have very, very limited visibility into the actual host cell protein in your product. Most companies that have tried this think that they probably have, 3 to 10 host cell proteins in their DS. But in reality, that number is typically, 20 to 100, maybe even more. But the image on the right is post-AAE. So you can see with our AAE column, it acts as an affinity column for host cell protein. So most of the product-related protein passes by, and we can enrich the host cell protein. So again, for the first time in our industry, we can give you a very realistic idea of exactly what host cell proteins are carrying through to your drug substance, which will allow you to perform a much more meaningful risk assessment to determine if the purification process development was successful, or if you need to maybe make some modifications before you get much farther. So this is just a quote from BioForum. BioForum is a consortium of industry leaders. They cover a very broad range of topics for development, but specifically for host cell protein. And I wanted to share this quote just to demonstrate that this is not just Cygnus saying this. This is the opinion of global biopharmaceutical leaders with respect to LCMS and host cell protein. So I'll just read a little bit of this quote here. It is recommended to perform HCP profiling by mass spectrometry as early as possible to guide process optimization at each stage of clinical development, leading toward commercialization to reduce high-risk HCPs to a level that ensures patient safety. So again, it's important to understand and control HCP to the best of your ability, and mass spectrometry is the best way to do that. So let's talk about some more of the chapter 1132-1. This is, again, host cell proteins that's specifically focused on mass spectrometry. So here's a couple of the just general characterizations of the chapter. It does acknowledge that currently there is really a lack of standardized methods. Again, just like I said back in 1999 with host cell protein in general, right now at the edge of the forefront of mass spectrometry, we are still kind of in a Wild West situation. However, this chapter kind of helps to work on that. It really gives us an overview of the capabilities of the methods, how we can do HCP identification and quantification, and specifically, and that's what we'll talk about here coming forward, is the limitations of these methods. We'll talk about best practices, or it does, for selection of your instrument, how you do your sample prep. It really emphasizes that mass spec and ELISA are very different. You can really go down some pretty deep rabbit holes if you try to correlate the methods or try to understand the differences or why they give you different results. So it's best to just not worry about that. And then how to make LCMS comparable between labs and expand our toolbox. So this is one of the tables that kind of It's a good way to summarize three approaches using LCMS to quantify individual host cell proteins. The first one, which was one that we tried at Cygnus a number of years ago, was quantifying individual HCPs relative to the product protein. What we found is that its very sensitivity was quite poor, its quantitative range was quite poor, but most importantly, its accuracy was quite poor. So we really pretty quickly deemed this as not a great way of trying to do individual quantification of individual host cell protein. The next is spiked in proteins. So here you can spike in a number of unrelated host cell proteins at known concentrations, and then try to measure the individual host cell proteins based on or relative to those. A much better method. It's quite sensitive. We get a 2 to 4 orders of magnitude for the quantitative range. The precision is very good, typically less than 20%. The accuracy is better than when it's relative to the product. But what we find is that it is not unusual to have orders of magnitude as in 2 to 4, 5, as much as 5-fold differences from the true concentrations. It's also moderate. You have to have pretty well described or defined standards, but it's not too terribly challenging. And this is quite decent for global relative quant. And then finally, spiked in peptides. So this is what we'll talk most about, or quite a bit about going forward. But this is spiking in individual peptides for each protein. Very high sensitivity, quantitative range is 1 or 2 orders of magnitude. But most importantly, again, for just quantification, the accuracy and precision are both quite good with this method. However, its one limitation is that it is very difficult to develop the correct heavy label peptides, and it can be quite expensive as well. So let's talk about the global relative quant. This is a Cygnus method and a service that we've launched last May called Inspect. So here, our approach to doing the global relative quantification is we have 8 individual host cell proteins that we have very well characterized and understand their concentration. You can see the different response curves of the individual HCPs in our method here. So again, this also points out why global relative quantification is challenging, because the host cell proteins can all, we talked about fragmentation, ionization, how they fly, can be quite different in their response. And that's illustrated here as well. But again, to give some way of doing the relative quantification, we take the median signal of these, which is kind of the red and orange there, or the red in the middle. And then we use that as our standard curve to measure the relative quantification of the individual HCPs in the samples. There is a part of that chapter USP chapter talks about NISTMAB. NISTMAB is a standard that has been sent out to a lot of different labs around the world to identify and quantify the individual HCPs. There's quite a few in the paper, but just for this example, we just took the top 4. So the OMIAL from 2023 has these top 4 HCPs, and just on average, this is quantification, and you can see our Inspect mass spec is on average pretty good. But again, as I said, when you're spiking in proteins or relative to proteins, 2 to 4-fold off is pretty normal. So you can see here again, it's in fairly good agreement with the published numbers. Here we've looked at accuracy and precision. So we've spiked in individual HCPs into a drug substance background. And you can see, again, these are relatively high concentrations, but they can be done in a way that's fairly accurate and precise. in a limited capacity. Okay, so let's talk about absolute quantification. This is by parallel reaction monitoring mass spectrometry. So again, this is that third part of the table. We're talking about spiking in peptides. So Cygnus released the 6X lipase assay again in May of 2025. This is specifically to address lipases. As I said before, we know that all host cell proteins are not created equal. Some host cell proteins are housekeeping, low abundance, low molecular weight proteins that really don't cause a problem. But some, specifically lipases, can be very problematic, even at extremely low concentrations. So when we're quantifying these proteins, having 2 to 4-fold or even 5-fold off really isn't useful. So we developed a method to give us a much more precise and accurate measurement of individual HCPs, again, because we know these could be problematic even at sub-ppm or even ppb levels. So again, this is a parallel reaction monitoring mass spectrometry. It's high resolution mass spec, unlike triple quad. It can provide a high level of specificity and sensitivity, and typically has a wider dynamic range than multiple reaction monitoring. One of the great things about this method is that its retention time is scheduled. So we're only scanning and measuring at exactly where the peptides that we're looking for are eluting, which also means that you don't have to have a lot of the upfront sample preparation or removal of the substance or even enrichment. So just by scanning that scheduled retention time measurements, it really makes it's a less modified sample and also can be done quite quickly. Here's our general workflow. So again, the sample is a typical processing, homogenize, reduce, alkylate. When we have peptides, we spike in the stable isotopically labeled peptides as well. Introduce those to the mass spectrometer, data analysis, and then the identification and quantification. This is an example of how this works. So you can kind of see one of the peptides here on the right-hand side. And again, these are stable isotopically labeled peptides. So they're going to elute off of the column at exactly the same time. They just have a slightly higher mass. So in this situation, we know the exact concentration of the peptides. So you can integrate the area under the curve and correlate that to the area under the curve of the same peptide from the unknown sample. So this is how we can get very, very accurate concentrations of these individual peptides, peptides which can correlate to the protein concentration. Here's an example of these, taking these proteins and spiking them at different concentrations gives a very linear response. Here's an example of making a standard of just one of the individual host cell proteins. This is the liposomal acid lipase. We made a 10-point calibration curve. You can see by the 5th point on the curve, we are both accurate and precise, so giving a pretty broad range of accuracy here. And more importantly, I'll show you on the next slide here, this is the quantitative range of each of the lipases that we have. So you can see again in the typically low ppm range, up to hundreds of ppm. On the right-hand side, you can kind of see an example of how we work through LOD, LOQ. So you can see in a blanket injection, there's no signal whatsoever. LOD, there's typically the heavy labeled peptide. Sometimes you can get a little bit of an incomplete peptide. So you can kind of see some of the trace. But LOQ, you can see the heavy labeled peptide and the different fragment ions below that. The more that you see there, that gives you the higher confidence that the method and the LOQ. So that's just kind of, again, how it looks at the very low levels of the quantitative range in this method. Here's a spike and recovery experiment using PLBL2 spiked into USP drug substance. Again, just illustration of accuracy and precision across the full analytical range of the curve. So you can see down, this one goes as far as 2.2 ppm. And finally, an example of the utility of this method and how individual biopharmaceutical companies can use it to help them either prevent problems or to solve problems, after they've occurred. So in this example, a company had sent Cygnus 2 different drug substance samples. These were totally blinded. We had no information about them going into this. Drug substance 1, you could see that we ran, and we detected 4 of the 6 lipases at reasonable concentrations. And when we talked to the customer, we found out that they had completely degraded the polysorbate 20 in 3 months of this sample. So again, it was, that was the case, the observed degradation of polysorbate, and you can see that it correlates with the reasonable levels of lipases across 4 of the 6. The second drug substance, we found only PLBL2. The others were all less than LOD. And the customer reported to us that there was no measurable polysorbate degradation in this DS. So 1, it shows the utility of our method, and 2, it gives further support to current literature suggesting that PLBL2 is not responsible for polysorbate degradation. However, it still is a highly immunogenic host cell protein. And here's a way that we've kind of put together to see the relative quant to the the relative quant to the absolute quant mass spec methods, the DIA, PRM. And it did this for a couple of reasons. You can see in the purple, you see the PRM method. This is the absolute quant of these lipases. They are both accurate and precise for all 6 individual lipase assays. The DIA method, again, is both accurate and precise for 1, but as much as 4 to 5-fold off for the others. So again, it's really a way to kind of illustrate that in the world of relative quant, it's great if you just need to get a ballpark idea of individual HCPs, but more importantly, if you want to correlate individual HCPs across samples. So if you're just looking as a comparison, is there more in this sample than this one? The relative quant might be a way to go. But in examples of lipases, proteases, and other host cell proteins that can be extremely problematic even at low levels, having this heavy labeled peptide PRM method is really important because it gives you much cleaner data that will drive you to make better, more informed decisions. Okay, so let's start wrapping up. So in summary, the LCMS is a powerful technique to identify and quantify individual HCPs. We found that the PRM with the stable isotopically labeled peptides is by far the most accurate. It is quite challenging to expand this to be a lot of individual proteins, so it is a more limited use for targeted problematic host cell proteins. But the DIA, again, if you want to get just a relative idea of individual HCPs, that's kind of a way to go. And we are currently working on new panels. Like I said, we have the lipase currently available. We're working on expanding that to proteases and then also other known potentially problematic host cell proteins. So just to kind of wrap up finally, all of the things that we've talked about in the webinar today are all services that are offered by Cygnus. As I mentioned at the beginning of the conversation, coverage analysis is something that we have become global leader in. We've done over 600 coverage analysis through 2D gels and mass spec that have been globally accepted and used for all filings, again, globally. We can identify the immunoreactive HCPs in your in-process samples as well as drug substance. So again, it's really good to look at this maybe into phase 2 prior to phase 3, so you can kind of make sure the process development is where it needs to be, and you can make any changes as needed. But also, if you do have a problem, if you change a process, if you scale up, it can help you understand where some problems might have happened. The InSpec mass spec, again, is a great relative way of getting the quantification, identification and quantification of individual HCPs. Again, best if used to compare across processes and not really of the quality of data for individual HCPs that should go into a submission, but it could be a good tool for process development work. And finally, the PRM mass spec is for absolute quantification. So again, this is when you need to have a very good idea of the exact concentration of problematic host cell proteins. Again, we currently offer this for our lipases, but we're expanding that to proteases and additional problematic HCPs as they become available. So where are we going? So we've really come a long way. Like I said, I've been doing this for 28 years, and we were at a point where there wasn't really any standard method. There wasn't any guidance. There weren't acceptance criteria. And we've come all the way to the point where we can identify and quantify individual host cell proteins. So where are we going, over the next number of years with this technology? So for the short term, I really suspect that, for process development work, and I think this is for the long term, process development work will probably always be supported with a generic or a platform ELISA. This is a tried and true method that has gotten us through almost 40 years of process development with very limited number of problems. It's also still relatively inexpensive and can be performed by analysts without a PhD. Lot release testing could be a little different. Again, for the short term, we absolutely will still do custom or generic host cell protein ELISAs. Something we didn't talk about today, but again, because we have some of these great new tools, specifically mass spec, we have the ability to know if a host cell protein ELISA is appropriate or not. So it is very common now for companies to just decide to stay with their generic or their platform assay, even going into commercial lot release. And I suspect it'll be that way for quite some time. We are a very conservative industry. I still talk to companies regularly about no longer performing TD Western blot. It's a method from the 1980s that really is shown to have some severe limitations relative to our new technology. But I suspect that for mass spec, it might have a similar acceptance curve to get over. But I can see long-term potentially mass spec getting to a point where it could be used for GMP lot release testing, again, at some level. The incorporation of mass spec now, again, coverage analysis, investigations, guiding process development, relative quant and absolute quant, that's just going to expand as we go and get to be a much higher level of importance. I can see even now we've had one example where there was a lot release test for HCP didn't have an ELISA available, and they were able to use mass spectrometry. But again, as I said before, at some point, again, it's hard to say with the conservative nature of our industry, as well as how long it's going to take these instruments to get to the level of shrinking the instrument, improving sample prep, improved AI data analysis that doesn't require PhD level scientists. But at some point, I think we can overcome a lot of these obstacles. And I would look forward to and expect mass spectrometry to be used in GMP lab release testing. Okay, with that, I'd like to thank a lot of the team. So Ala Zilberman is our VP of Business Development, Technical Marketing. Jared Isaac leads our chromatography and mass spectrometry team. Tim, Stefan, Johanna, and Brinkley are also members of that team and have generated a lot of the data that is in the presentation today. So with that, say thank you. And if we have any time available, I think we can take a few questions.
Great. Thank you, Eric, for that presentation. I do see some questions coming in, but before we get started on the Q&A session, I would just like to remind our audience how to submit questions. You can do so by typing them in the Q&A box, which can be found directly below the video player. Our first question for you, Eric, is if we perform AAE with LCMS for antibody coverage analysis from the F551 kit, will we be able to use this kit as our release method in BLA filing?
Well, unfortunately, the question is not quite as simple as that. Again, I often talk with our customers that to keep in mind that coverage analysis is simply one part of determining if an assay is appropriate or acceptable to use for BLA filing. But yes, if your coverage is good with AAE and LCMS, and that pairs well with the qualification or validation in the ELISA, you absolutely can use that method to justify the use of the assay.
Great, thank you. So our next question we have in is asking if you know of any cases where product was released using host cell protein quantification by LCMS only.
So as of now, there's only one example that I'm aware of. It happened during COVID, where a company was developing a COVID vaccine using a cell line that there was no HCP analytics available. It was approved on an emergency basis to do that. But to the best of my knowledge, that product never actually made it to the market. So there was at least approval to take that route, but it was a product that ultimately was not successful.
Okay, and then our next question, when should inspect MS be used and when should the lipase MS assay be used?
Yeah, great question. So yeah, these are two very different methods that have quite different, but also very useful applications. So the inspect mass spec, again, so that's a relative quantification. So if you're trying to compare HCP profiles across two runs or two processes, it's a really great tool because you can use it to identify and determine a relative quantification of each of the individual HCPs. So for instance, if you are producing a biosimilar and you want to compare it to the original drug, maybe the inspect is a great tool there. Or if you're just looking at, you ran a process in your development lab and you transferred it to your CDMO and the HCP seems to be higher than it was before. Using the inspect can give you an idea of exactly which ones are there and if something's being enriched or removed. So it's a great way to compare HCP across processes on an individual HCP level rather than just the total MS. The lipase MS assay, again, it's done more as like a verification before you launch your purification process so that you can make sure that you're not going to have any lipases present at concentrations that are going to prove to be problematic downstream. So again, you really don't want to wait until you're 3 to 6 months into a stability study to find out that you have a lipase problem. So it's a really good idea to use that early before that. But if you do find yourself with a problem, again, that method is a great tool to help you identify what lipase is present, and that can inform the process development team of ways to work on removal of those individual lipases.
Great, thank you. I think we have time for one more question before we start to close out. So to wrap up, can we purchase an AAE column?
Good question. Thank you. So currently, Cygnus offers AAE as a service. Again, it's a proprietary method that we've developed in-house using our columns. So currently, we do only offer it as a service. If you're interested in our AAE and especially our mass spectrometry services, we really encourage you to reach out to our business development team or technical support team, and we can help get you in touch with the right people and make sure that do a project to answer your questions or meet your needs.
Thank you. All right, with that, I do think it's time to already start wrapping up. So first, Eric, thank you for presenting today and answering those audience questions. And thank you to the audience for attending and participating in today's event. I'd also like to thank our sponsor, Cygnus Technologies, for making today's educational webcast possible. We'd also like to ask everyone in the audience to participate in a brief survey, and that's going to be appearing on your screen after today's presentation has ended. You'll also receive an email alerting you when this webcast will be available on demand for replay, and we invite you to forward that announcement to any of your colleagues who may have missed today's live event but still want to tune in. Otherwise, we hope to see everyone next time. Thank you and take care.